Just as we learned very early in this course, I think in lesson 3, structure is function, not only for proteins in our cells, but also in the world of pharmacology. Remember that drugs bind to proteins in your cells and in your body. And if a protein has even a slight change in its conformation or structure, it will likely change its ability to interact with the drug or the speed with which it can interact with the drug. Structural variation in pharmacodynamic proteins affects the target effects of a drug. For example, the drug here in red binds the receptor here in this Y-shaped conformation. When it binds, there's a normal activity that happens, a target effect, if you will. But if we have a variation in the structure of this receptor, now the drug can't bind to it in the same way, so the activity turns out to be reduced. A genetic variation that altered the structure of the protein resulted in a functional change for the drug. Structural variation in pharmacokinetic proteins affects the speed with which a drug is metabolized or broken down. In this case, the drug is in red, being metabolized by the purple protein into these two smaller metabolites. This would be our normal drug metabolism. But we could have a variant over here that changed the structure of the enzyme that breaks down the drug. And now there's a reduced activity or no drug metabolism at all. Let's see if you can pull on the information we've learned about up to this point in the lesson. What do you think would result from a structural change of the GABA receptor causing an increase in the pharmacodynamic efficiency to bind alcohol, and a structural change of alcohol dehydrogenase or ADH causing a decrease in pharmacokinetic efficiency of alcohol metabolism? Would you expect extreme intoxication, moderate intoxication, or minimal intoxication? Remember, this time we're combining what we know about the pharmacodynamic and the pharmacokinetic pathways at one time to see the overall effect of the drug on your whole system or your body.